Today I'm going to be talking about evolution of dispersal and plastic generalism during range expansions. First off, I would like to acknowledge my PI and co-author on this work, Jeremy Draghi, as well as other members of the Draghi Lab for a lot of helpful feedback on this work. So I'm going to be talking a lot about range expansions today. When I say range expansion, I'm referring to a population moving into a new area that doesn't already have conspecifics in it for the first time. And thinking about range expansions is particularly relevant right now because of invasive species all over the planet and also climatic shifts potentially leading to populations needing to shift their geographic ranges. During range expansions, evolution can get kind of weird. One example of this is with allele surfing. With allele surfing, alleles that are present on the edge of an expansion have a higher probability of success than expected by their fitness in a stationary environment alone. So this is because along the expansion front, there's a smaller population that's being sampled from, and so there's a higher chance of genetic drift there. This also means that selection can be weakened during expansion, where fitness that an individual would have in a stationary population doesn't necessarily reflect what its success will be in an expanding population. This also means that positioning is much more important during range expansions than in a stationary population. Where an organism is positioned isn't only random, though. Traits like dispersal can also impact where an individual is going to be located. We see during range expansions that there can be evolution of higher dispersal along the expansion front. So as we see here, this is a simulation of a population starting with no standing genetic variation, and we still see evolution of longer dispersers as the expansion continues along the range front. We also see this in empirical systems. So in this figure from Shine et al. 2021, we see stationary populations on the left side of the graph of cane toads in Australia, which is a really famous example of dispersal evolving during expansions. And then on the right side of this graph, we see an example of a range expansion. And we see that populations that are closer to the range edge have more displacement per day than the populations that are closer to the core. So if we're seeing dispersal distance evolving during range expansions, this can also impact other things in the population. One thing is the level of heterogeneity that a population will experience. Throughout this talk, I'm going to be talking about computer simulation models that I've done. And in these models, we're looking at individuals that are stationary throughout their lifespan. They randomly disperse their offspring, and then their offspring are stationary. We also had a landscape that had two different environments present in clusters. So if we have this focal individual in black, if it's dispersing a short distance, we see that a lot of its offspring are going to disperse to the same environment as the parent. Whereas if we have longer dispersal, we see that more of its offspring are going to disperse to alternate environments. So what this means is as dispersal distance increases, we expect that experienced heterogeneity is also going to increase. When we think about how heterogeneity could select on niche breadth, or the amount of environments that an organism is adapted to, we can think about specialists are going to have lower fitness as the heterogeneity increases. As heterogeneity increases, they're going to spend less time in environments that they're optimized for, whereas with generalists, we expect that their fitness is going to stay more consistent across a range of heterogeneities. Here we're seeing a costly generalist where in low heterogeneity it doesn't have fitness that's close to the specialist, but as we reach higher heterogeneity, we see where the benefits of generalism are more important than the costs. So putting this all together in a range expansion framework, we know that during range expansions, dispersal distance can evolve to increase, and this can increase the heterogeneity that randomly dispersing populations will experience. We also can see that when populations experience higher heterogeneity, the benefits of generalism can become more relevant than the costs. So the main question that we are trying to explore here is, can increased dispersal during range expansion lead to increased heterogeneity and selection for generalists even when they're costly? So for this, we modeled a landscape with two different types of environments, and organisms had a single mobile juvenile stage and then were stationary for the rest of their lives. In this model, our organisms had a couple different traits that it could evolve that we're largely grouping into a dispersal trait, which determines the dispersal distances of the offspring, so what that distribution of distances looks like, and then niche traits, which were determining an organism's fitness in the environment that they're in. With these niche traits, we had non-plastic traits, which had a constant contribution to the phenotype, and plastic traits, which had an environmentally dependent contribution to the phenotype. So to understand a little bit more what our fitness landscape looked like here, we have here a range of phenotypes on the x-axis, their fitness on the y-axis, and then the lines represent the distribution of fitnesses in each of the two environments. So our specialists are at the optimum in each environment, where our generalist that's being produced just with our non-plastic contributions to the phenotype is occurring in between these two different optimums. And we see that while it has decent fitness in both environments, it doesn't have as high fitness as a specialist in either environment. We also have our plastic trait, which can change the phenotype from what's created with just our constitutive non-plastic contribution. So here we see cues from environment A would push the phenotype in one direction, and cues from environment B would push the phenotype in the other direction. We can also see that the generalist's fitness is can increase with plasticity, whereas specialists, which were already at the optimum in a given environment, are going to be pushed away from that optimum by increased sensitivity to a cue. So in our models, we started with an ancestral organism that was a non-plastic specialist, and it could have non-plastic mutations, which are going to produce a jack-of-all-trades, master-of-none generalist, and we expect those to primarily be favorable in periods of high heterogeneity because of the cost that they have. We can also have plastic mutations, which we saw are deleterious when they arise in specialists, but are beneficial and can increase the fitness in generalists. And both of these types of mutations can come together to potentially create a plastic generalist where when both mutations co-occur, we get a well-adapted generalist. And what this means is the generalist is still going to have consistent fitness across low and high heterogeneity, but the fitness of it is going to be higher across all levels of heterogeneity, and the area of heterogeneity where the benefits of generalism are more relevant is going to be larger. So we expected that with these two different types of mutations, because the non-plastic mutations would be favorable in high heterogeneity, potentially we would have an increase in frequency during range expansion and increased dispersal that would provide more individuals where plastic mutations could be beneficial and potentially change the long-term behavior of our populations. So first, when in these simulations, we saw that dispersal distance did evolve to increase, so that part seemed to be consistent, where dispersal distance increases throughout range expansion, and the end of range expansion is going to be shown by these vertical dotted lines. So once range expansion is over and those increased benefits of dispersal is over, we saw dispersal decrease. When we looked at the frequency of generalists, we saw that when there was just a stationary population without range expansion, the costs made so we didn't get a lot of generalists arising. When we modeled a population that didn't have plasticity, so it didn't have the ability to reduce the costs of generalism in this population, we see that the frequency of generalists increases during range expansion, and it continues to increase a bit after range expansion while dispersal is still high, but as dispersal decreases, we see that it's also decreasing. And when we incorporated plastic mutations, which allowed generalists to reduce the cost of being a generalist, we see that the frequency of generalism increases during range expansion, decreases a bit, but continues to increase because it seems like they're able to then find plastic mutations and reduce the cost of generalism. So far, this seems great with the chain of thought that we thought was producing this behavior, where we have range expansion leading to increased dispersal distance, leading to increased heterogeneity, and then increased frequency of generalism and an increased chance to find these cost-reducing plastic mutations. And if this chain is completely correct without any complications, we would expect that if we take away the ability of dispersal distance to evolve, that then we probably wouldn't have this increase in heterogeneity and wouldn't have an increase in the frequency of generalists. That ended up being not what we saw. So what we see here is in the teal line, we have constant dispersal, where dispersal is held at a relatively low value throughout the simulation, whereas the purple line is that evolving dispersal and plasticity line again. So basically what this means is we know that range expansions seem to still be leading to an increase in the frequency of generalists, even when dispersal isn't able to evolve. So in the stationary population, we didn't see an increase in frequency of generalists, but during range expansion, even without dispersal distance evolution, we have an increase in the frequency of generalists. And when plasticity is present in that situation, we see that it also seems to have a long-term impact on the population. So right now, we honestly don't totally understand what's going on here. There's a lot of complications, and kind of every simplified test that we've run so far has been contrary to our expectations. So it's really exciting right now to kind of try and understand what other benefits of generalism during range expansion are happening. A couple of things that we've tested are, as you can see, the end of dispersal in the dotted line in the teal is later than the end of dispersal, or the end of range expansion on the purple line. So we thought maybe the longer period of range expansion could be allowing generalists to reach higher frequencies. When we controlled for the length of time that range expansion was happening, that also did not explain it. When we were controlling for the difference in fitness due to different levels of heterogeneity with constant different dispersal distances, that didn't explain it. And so we're really excited to look more at the ecology of this system and try and understand what things might be happening that are even more contrary to our expectations than we expected. So the main conclusions here are that plastic generalism seems to be beneficial during range expansions in even more ways than we hypothesized. And also that even though this study is using simulation models where we created this model and set the parameters, we can still get really interesting emergent behaviors. And with these models, we're then able to simplify back to the ecology to really try and get at the mechanism. And now I can take any questions. Yeah, so the plastic mutations that I was showing were just kind of an intermediate value, but they are able to match the fitness of the specialist if they have a large enough plastic mutation. Mm-hmm.
I'm just curious about the relationship between the sort of spatial scale of your dispersal distances, but also like the grain of the environment. So if the grain were finer and the core dispersers at the core are still encountering those differences, would you expect that you'd see more generalists across the board?
I think so. I mean, I hesitate to make too many predictions just because we're seeing that there's a lot of complexity that isn't matching our predictions. But this kind of emerged, this study idea emerged from originally looking at grain sizes and then kind of exploring how dispersal could change. I think the shape of how dispersal distance would relate to heterogeneity would change, and that could change things. I would expect that that generalism would be generally more favorable. But yeah, we aren't totally sure what that will look like yet.
So hi everyone, thank you to be here. I'm very proud to doing my presentation today because this is my first year doing a PhD, so it's a huge thing for me. So just a few words before my presentation. So my name is Théo Chavanis and I work on mountain butterfly and especially on pressure of selection, like the predation and the climatic condition. Today I will present you how the butterflies can answer against the climatic condition. So for the beginning, I just want to present you where the study is localized. So we are in French Alps. What you need to know in the mountain ecosystem, that you have many gradients. You have obviously the elevation, and along the elevation you have many ecological gradients. You have, first of all the gradients linked with the interaction, like the predation, as you can see by the bird, or for example by a crab spider. And you have also other interaction, like the plant composition, and also for the butterfly, for example, the competition between the pollinizator. And you have other gradients like the climate condition, for example, the temperature and the precipitation. And in the mountain ecosystem, the climatic condition growing very fast. Indeed, you have at high elevation, you have a strong precipitation with a colder temperature, and at low elevation, you have a drier condition with a warmer temperature. And butterflies are very interesting because they are very sensitive to this climatic condition. And indeed, there are ectotherms, and ectotherms are they have activities conditioned by their thermal requirement, and their survival are very limited by their thermal tolerance. And as you can know, and I hope, butterflies have wings, and wings have a huge role in the thermoregulation. Indeed, they can, with microstructure and colored pigments, dissipate the heat when they are overheated, and also they can focus the solar energy when they are in a colder condition. And one hypothesis, very interesting, and work on relation between the coloration and the climatic conditions. This is the thermal melanism made by Boger in 1949, who said that in the colder climate, you have a darker population, and in a warmer climate, you have a darkless population. And if we put this hypothesis on the mountain ecosystem, you're going to have potentially, at the high elevation, a darker population, and then at low elevation, a darkless population. And so that's the first question of my PhD. That's to know how have the coloration of butterflies evolved in the relation to altitude and climatic conditions. And so for that, first of all, I needed to create a data set to know where the species of butterflies are localized on the French Alps. So for that, I use a huge database in France that called INPN, which is a naturalist database for the observation. And I found, first of all, 500,000 observations for 230 species. But maybe you know, for this database, there is some data which are replicated, and also there is sometimes wrong information for the identification, but also for the localization. So I did a treatment for this data that I found, and finally I've got a data set with 175,000 of observation for 192 species. And with this observation, I was able to have the elevation and also the GPS points. And with this GPS point, with another database focused on the climate data, I was able to extract the temperature and the precipitation. And what is very important, this is the precision of the climatic data, because for this database, I don't call the data for 50 meters, because in the mountain ecosystem, around, for example, 1 kilometer, if the climatic data is not precise, you can be at the top of the mountain or at the down of the mountain very, very rapidly. And so after I did this data set, I did another data set, and this time for the coloration of the butterfly wings. And for that, I went to the Paris Museum and on the Lepidoptera collection, and I collect all the species that correspond to my other dataset. We can see a sample here from the collection. And I'm taking lots of photos. And with these photos, I was able to extract the reflectance. And it can be correspond to the absorption. As you can see with this equation, very, very simple. We admit that the transmission of the wings of butterflies is equal to 0, so we can say that potentially the absorption is equal to the opposite of the reflection. And the absorption, this is what we need to know for the relation between the coloration and the climatic condition. And so I'm taking photo for, it's not working anymore. I don't know if, yeah. So I'm taking photo on ventral view and dorsal view because maybe, as you know, the behavior of the butterflies is very different in function of the species. Some species need to have the wings open to thermoregulated, and some species need to have a wing closed to doing thermoregulation. And I'm taking also separately males and females in photos because, as you can see, there is sexual dimorphism for the coloration, and some species are not the same coloration for each gender. So, and after I'm taking photo for the UV color, for the visible color, and also for the near infrared color until 2,500 nanometers. And finally, I've got a dataset with 4,344 photos. And with these two datasets, I was able to verify the thermal mechanism hypothesis with some analysis, like a PGLS with a phylogenetic hypothesis, phylogenetic correction, with a relation between the coloration and the elevation or the climatic condition. And I take also the size in this relation because the size is very important for the thermoregulation. If you have a bigger butterfly, he absorbs not the same solar energy than a smaller butterfly. And so after that, I've got some results. First of all, I did a relation between a species richness and an elevation. And as you can see, there is 3 stages. There is one stage that corresponds to a threshold between 800 meters and 2,000 meters, so where the species richness is very stable. And there is 2 other phases, one before 800 meters, that corresponding to a decreasing of species richness due to potentially of the urbanization. And there is another phase after 2,000 meters that corresponds to a filtering by elevation, and where we see a decreasing in species richness potentially due to a climatic condition. And so after that, I did the relation between the reflection and the elevation. And we could see here for the dorsal view and ventral view for different type of color that we have a darker population in a high elevation. So it means that potentially the species which are in a high elevation can absorb more solar energy. And we can see, especially for the infrared color, that is very significant. And this is why? Because this is because the infrared coloration have a role in the absorption of the solar energy. And we saw that the same thing for the relation between the reflectance and the temperature. And we see that there is a darker population on the cold climate. And also this we can see, so there is, it means that species at cold climate can absorb more light than other species. And we can see also there is it's very significant for the infrared, which have a role in the thermoregulation. And so we can conclude briefly that there is a filtering by altitude from 2,000 meters. So we can potentially know that there is some species that are very specialist for a high altitude. And we can see also there is a thermal mechanism observed for infrared colors. And that's important because some other paper and also other research have found the same result, but for visible color and infrared color. But if we localize for a mountain ecosystem, we can see that it's just for the infrared color. So it means that thermal advantage has a role in the evolution of butterflies coloration in the Alps. And so my first work, because it is just an introduction of my works, have some perspectives. First of all, to know if the optical properties are linked to the thermal properties for the butterflies, to know if some butterflies can warm better than other species. And there is another perspective for the response against the climatic conditions, to know if the butterflies are adapted to their environment. So it means that if there is some species that can be more tolerant to a colder climate, and some of the species can be have a better thermal recruitment for their climatic niches. And another perspective, but this is just another work, but to know if there is another answer to other selection, like the pressure. And we did already some work on the field with the experiential predation with bird and spider crab and also grasshopper. If you want to talk predation, I will be open for that also. And yeah, I just want to thank Marianne Elias and Anne Michelin, who are my professor PhD, and also Colin Fontaine, who helped me a lot for the data selection and the analysis. And thank you for your attention. Yeah.
This might be a little off topic, but can butterflies respire at really high elevations, or do they have problems with actually breathing?
Yeah, maybe, but we see there is some species like Arabia. I don't know if you know it. Yeah, it's so a dark butterfly, which live in until 3,000 meters. But so I don't think there is a problem for the respiration. But if we think the relation between the species richness and the elevation, we think that until 3,000 meters, there is no more species. So yeah, I think maybe it's a parameter for the known there is no butterflies at high elevation. Yeah. Yeah.
Do you think also like the gravity field or like the species richness of plants will affect the gradient you have, and therefore it's an indirect correlation you observe, not the direct one?
So with the plant composition, that's.
Or just the richness of species
Yeah.
plants you have in the population.
Ah, yeah, okay. Yeah, I don't know. Maybe I didn't thought about that. I think it's a good question, but yeah, I didn't think about that. Thank you.
Hello. I'd like to talk about the coevolution of species range and niche in changing environments, which is both a classic topic and it's obviously relevant as we are facing faster changes in the temperature and other conditions. And in particular, I'd like to talk about the importance of gene flow, and I will talk a little bit about plasticity as well. So what constitutes a range margins is obviously an old question, and one of the influential comments on that was written in 1957 by J.B.S. Haldane, who explained that you would expect to have quite a different type of selection at the range margin to the center of the species range. And obviously the range margin, by definition, you would expect there's going to be somewhat lower density, right? That lower density is going to lead to an asymmetric gene flow, that asymmetric gene flow is going to increase the relative maladaptation and the range margin. And that is going to, again, increase the gradient in the density. It's going to make the maladaptation gene flow higher, and it could, in principle, lead to stable range margin, right? And that's been popularized much later as the swamping gene flow idea. Halde didn't actually do any modeling on that. That came much later. One of the influential quantitative models of that goes back to Kirkpatrick and Barton in 1997, who explained that the effective environmental gradient increases with dispersal, and as this gradient steepens, species need higher genetic variability to adapt continuously. And if that variability is too small, relative to that effective environmental gradient, you get a sharp range margin. Okay? So that seemed to confirm that idea that Halden had in the 50s. The problem with that is that genetic variance is not fixed, it changes, and that can affect our conclusions and our understanding what constitutes a stable range margin. Okay? So gene flow obviously has multiple effects of adaptation. As I alluded, it increases genetic variability that will increase local average maladaptation. But if the optimum, so here you see in blue some sort of distribution of some toy trait mean, and the red line gives you some optimum, right? And so if that optimum moves in space, if the optimum moves in trait space, all else equal, the species that has a higher genetic variability will be closer to that optimum, right? That's a standard classic lambda equation. The other effect that gene flow has that's relevant for evolution of species range is that the neighborhood size is going to increase with gene flow and dispersal. And I say neighborhood size, I actually mean neighborhood size, not effective population size. Neighborhood size goes back to Wright in the 40s, and it measures the strength of genetic drift in a spatially continuous populations, and is particularly relevant to the ability of the population to maintain diversification across local scales. And you can think of it as the number of individuals that's available within one generation's dispersal range, right? If you have more gene flow, you get higher neighborhood size, and you get higher efficacy of selection. So there's quite a non-trivial feedback there, right? What I will do, I'll present a simple model that assumes that there is a single trait or compound trait that's changing in one dimension, say latitude, could be also altitude, and there is another dimension where that trait is neutral. So it can be longitude, or if you think of a hill, right, you're going to have stuff changing with the height of the hill, but not along the hill, okay? And that optimum is changing through time, say like so. What's important for this type of model is a coupling between the mean fitness of a population and that reflects the adaptation and the growth, right? And so we need some evolution of an adaptive trait, some evolution of genetic variants that are going to evolve while changing allele frequencies, and these are going to change due to dispersal, selection, stochasticity, mutation as well, which I skipped. If you don't like these, just ignore it. For the people that are familiar with the framework, there is some evolution. Here I show evolution of allele frequencies, assuming the linkage equilibrium. There's dispersal in blue, selection in green. There's some mutation and some stochasticity in purple. There's some ecology, which is again dispersal in blue, and some growth. This is the Rn term, right? And there's some stochasticity. Decoupling comes via the mean fitness R bar, and it has a density-dependent component. This is the R times 1 minus n over k, and it has some cost due to stabilizing selection and some cost due to genetic variance. Okay, can't forget about these. So I did run a lot of simulations, and I just showed an example of these, right? And I would like my English doesn't work. And so in the top left corner, you see what the optimum could look like. There's a trait that's matching in blue. On the top right, you see the population density, and then in the bottom, I guess in your case actually, right, are the allele frequencies that are quite messy. Some of them are less messy than others, and you just see a sample of them, okay? And so you can maybe focus on the black one that's changing in some sort of slightly noisy way from frequency 0 to frequency 1. And it's each of these alleles changes from 0 to 1 that contributes alpha to the trait mean, and that alpha is drawn from some exponential distribution. And then you've got some genetic variants, and we have some approximations of that. The dashed lines, the black gives the deterministic expectation, the green gives the expectation with drift that I will not explain. Okay, and so as optimum changes through time, the population is going to move and adapt, right, in general. And so you see the movement of the population, you see the movement of declines, and there's some adaptation, but it's quite minimal in this particular example. Okay, so that's a simulation, and it's not very useful, right? It's just to illustrate how the model looks like. What is useful is that if you remember these, say, equations from earlier on, they allow one to figure out what the dimensionless parameters are that describe the system. And so one of them is the effective environmental gradient, which is what's the cost in fitness of the heterogeneity. The other one is the effective rate of temporal change. Again, what's the cost in fitness that's imposed by the rate of the changing optimum through time. There's the neighborhood size, that is relative strength of selection, and that is mutation rate. In the absence of temporal change, the first two parameters are important to predict stable range margins. And I addressed this earlier, and here what you see is two of these parameters, the neighborhood size on the y, and the effective environmental gradient on the x. And the dots, each dot is a simulation, and the parameters are drawn from this, 10-dimensional parameter space, assuming we do not know the scaling. And the blue populations are expanding, the red ones are collapsing, and you see there is a decent separation between the populations that are happy and expanding on a linear gradient, populations that are not happy and collapsing. The open circles give you fragmented populations. What this means is also, as you're moving through the parameter space, say you can move through the parameter space with a hidden population by steepening the environmental gradient, then you would move from the blue to red and you would get the stable range margin. Okay, so it doesn't necessarily imply that we think that populations live on a linear gradient. It's just a simple way to run a lot of simulations and make some sense of them. So we know what happens, well, we have some understanding what drives the dynamics in the absence of temporal change, right? But we obviously want to know what happens with temporal change. And that gets complicated because it's hard to draw in more dimensions. And so what you see in here is the same coloring as before, but now neighborhood size is fixed, say to roughly 15. You've got the same effective environmental gradient on the x, and you've got the rate of temporal change on the y that's scaled in terms of fitness cost. And the color give you the rate of collapse in yellow and red and the rate of expansion in blue, right? And you see that there's some patterns, but it's hard to make sense of it, right? Now, what you see with the crosses are the populations that went extinct within 500 generations. And what you see now is a simpler prediction assuming genetic variance was fixed to what's implied by the spatial heterogeneity and didn't change through drift or temporal change, okay? And so that gives you some idea, right? I mean, it's certainly better than nothing. One thing I didn't point on this graph earlier is some of these populations are fragmented, so I do it right now. So these are fragmented such that 30% of these range is not occupied, and that's an arbitrary threshold, okay, bear that in mind. And I show in this green arrow, so that green arrow in there, that's the dashed line that you saw on the earlier chart, if you remember. Can I go back? No, I can't, right. There was a dashed line that was separating the blue from the red, and that's the green arrow right here, right? What we want to know is how this changes with temporal variation, right? And so what we can do, we can do a simple correction of that threshold with how much more of a load would we expect because of the predicted lag load, correct it for the expected genetic variance with genetic drift. Now, that expectation of the genetic variance with genetic drift is partially heuristic, right? So I know what the shape of that function is, but I had to fit a constant numerically in the absence of temporal change. And so there is certain uncertainty about where exactly this dashed curve is, but it's roughly somewhere there. Okay, so that gives us some idea about if the population is expanding or contracting. I've got 1 minute. That's great. So genetic variance evolves with the temporal change and with the spatial change. The change of genetic variance due to spatial variation then enables adaptation to faster temporal change. And I will not tell you anything about that, all that. Gene flow will make adaptation easier. If I change the effective environmental gradient by increasing dispersal, that is going to increase neighborhood size. The increase of neighborhood size is more important than the increase of the gradient. That will make adaptation easier. Okay? Plasticity doesn't matter really that much. You can ask about it. Right? The summary is the dispersal across spatial gradients enables adaptation to changing environments by increasing genetic variance that enables higher rate of adaptation, by increasing neighborhood size that lowers genetic drift. And with fast temporal change, the cost of increasing standing load may be less important than the gain in the rate of adaptation. And that threshold is quite robust to adaptive plasticity. I can get back to this in the questions. Yeah, and I'll be happy to welcome questions, now or later. Yes, please.
I was curious about the effect of plasticity because normally in those models it's changing the slope of the
Yeah, so you would think, right, that plasticity would just rescale the stabilizing selection, but the problem is that there's evolvable variance, and so when you have plasticity and evolvable variance means the variance is smaller, and they seem to cancel out quite nicely. But maybe that's up to further scrutiny. But the simple scaling is obvious, right? But once you have evolvable variance, then things work slightly differently. Am I making sense? Plasticity, in my case, it's very simplistic, and it just means that it's adaptive plasticity, so the species trait is closer by certain constant to the trait optimum than the genetics.
Hi, everyone. I'm Chris Carlson. I'm a PhD student at the University of Toronto. And today I'm really excited to be talking to you all about how G/E interactions can maintain variation in mutualisms. All right, so why should we care about genetic variation in mutualisms? Well, one reason is that having really diverse partnerships can be more robust in the face of changing environments. However, mutualistic interactions often co-evolve via genotype by genotype interactions, which occur when the fitness of a genotype in one species depends on that of its partner. So here as an example, in this interaction between this hawk moth and an orchid, and this focal interaction, both genotypes match and receive a large benefit, but in an interaction with this orchid with a massive nectar spur, the hawk moth is unable to pollinate, and neither partner receives a benefit from mutualism. And when these interactions are mutually beneficial, it can lead to positive frequency-dependent selection, which we predict in simple models will actually erode variation within mutualism, as a pair of matching partners fixes within the population, outcompeting the others. And I think this begs an interesting question of what might maintain variation in mutualism and make them more robust to things like global change. So one explanation, one factor even, that could maintain variation in mutualisms is that these interactions occur in already quite heterogeneous environments. So in a toy example, consider the state of California. California's Central Valley is quite different from the nearby Sierra Nevada mountains. And as organisms experience this heterogeneity while dispersing, this could maintain variation within mutualisms. So these heterogeneous environments can give rise to genotype by environment or G by E interactions, which occur when different genotypes are plastic in different ways, right? So here, genotype 1 does well in environment 1 and poorly in environment 2, but genotype 2 has an opposite reaction to the changing environments. G by E can maintain variation in heterogeneous environments where some genotypes assort to their preferred environment and others to their preferred environment, and dispersal can lead to fluctuating selection dynamics that maintain variation. However, within mutualisms, G by E is more complicated because we have to think about it in not just one, but two species, and the way in which these species disperse can be asymmetric, meaning they disperse at different rates. And so, given all these complications, when do we expect G by E to maintain variation in mutualisms? You know, I couldn't tell you off the top of my head, so we built a model to figure it out. Okay, so here's an overview of the model where we had two host and two symbiont genotypes. In the model, fitness depended on the environment. So here we have host 1 and environment 1, where it could interact with symbiont 1 or symbiont 2 in proportion to their frequency. When interacting with symbiont 1, host 1 is well matched to its partner, the partner is well matched to the environment, and the host is matched to the environment. So it receives a fitness payoff according to a perfect match. But in this alternative interaction, host 1 is mismatched to symbiont 2, and symbiont 2 is mismatched to its environment. So it gets a payoff from just matching its own environment. The model featured two distinct environments between which the hosts and symbionts dispersed, where they could have the same set of interactions. And for generality, we arrived at symbiont fitnesses in the exact same manner. All right, so to ask how this system would evolve, we modeled the change in frequency of each genotype, such that each genotype increased in frequency when it was fitter than its competitors in a particular environment, and the genotypes dispersed into and out of each environment. All right, so the model exhibited 4 distinct outcomes. The first outcomes were fixed, where just one host and symbiont fixed globally, either in a global genotypic match or a global genotypic mismatch, where mismatched genotypes fixed everywhere. There were also polymorphic outcomes where variation was maintained within each environment, either via migration selection balance, where matching genotypes are frequent in their preferred environment, or via a reverse migration selection balance, where matched genotypes were frequent in their mismatched environment. And we found that which outcomes our model exhibited depended on the relative strengths of G by G and G by E interactions. So to show you what I mean by this, when G by E was weak in our model, it meant that if we consider the fitness of the host fixed for just symbiont 1 while looking at environments 1 and 2, that host 1 always received a high benefit from symbiont 1, but symbiont 2 always did worse with symbiont 1. And these two genotypes are not responding very differently to either environment. Alternatively, when we think about the strength of G by G, by considering change in fitness across either symbiont type, only in environment 1, we see that the two host genotypes respond quite differently to the symbiont. We found that there were 4 scenarios reflecting the strength of G by E versus G by G that determined the outcomes of our model. G by E could be weak for both partners, intermediate for one or both partners, very strong but in just one partner, or very strong in both. And I don't have time to go through all these scenarios today, but I will talk about scenarios 1 and 4 at the extremes. All right, so we did all this analysis. What did we find, and what does it tell us about variation in mutualisms? So first, when G by E was weak for both partners, we found that there were 3 evolutionary outcomes. The first was the global genotypic match, which was stable for any level of dispersal, or migration selection balance and reverse migration selection balance, where local polymorphism was maintained. So we saw that these equilibria were stable for any level of dispersal. And importantly here, we see that 2 potential outcomes are polymorphic. This suggests that even when G by E is weak, that population structure and frequency dependence can maintain local variation in mutualisms. All right, so now on to scenario 4, where now we see that G by E is very strong relative to G by G for both partners. So here, we found that there are 2 evolutionary outcomes. Either the global genotypic mismatch, which is stable when dispersal was high, or migration selection balance, which could occur for any level of dispersal. So it makes sense that when matching the environment changes rank order fitness to a high extent, that we might get this migration selection balance outcome that reflects, the high benefit from matching the environment. But why would we ever get this outcome where we only have a pair of mismatched partners, where G by E is essentially eroding variation? The reason comes down to the high dispersal in this case. So when we think about the fitness of just host genotype 1 between environments 1 and 2, when interacting with symbiont 1, host 1 pays a high fitness penalty from being in environment 2 with that symbiont, because the host mismatches the environment and the symbiont mismatches its environment. However, when interacting with the other symbiont 2 still does quite well in environment 1 and can provision a high benefit to the host. And so on average, with high dispersal, it means that it's actually better to have this mismatched partner in a well-mixed environment. So this result suggests that strong G by E in both partners can erode variation when dispersal is very high and under the right initial conditions. All right, finally I want to talk about the role of asymmetric dispersal rates in our model. So what happens when hosts and symbionts disperse differently? In broad strokes, we found in this case that the more slowly dispersing partner could be rate limiting with respect to those fixed outcomes of our model. So let me go through an example to explain what I mean by this. So let's return to back when G by E is strong. Here, we would normally observe the global genotypic mismatch or migration selection balance. But let's say if the host disperses very slowly and the symbiont disperses a lot, we find that if the host disperses slowly, it doesn't matter how much that symbiont disperses, we can never reach the global genotypic mismatch. The slowly dispersing host prevents it. And so this result suggests that asymmetric dispersal can create a two-body problem in mutualisms that tends to increase variation in scenarios where it would otherwise be eroded. All right, so what did we learn today? I presented a model where I was steadily increasing the strength of G by E interactions in mutualism. I found that when G by E was very weak, that variation can be maintained or eroded irrespective of the level of dispersal. But when G by E was strong, we found that it always maintained variation at low dispersal, but but it could erode variation as dispersal became high enough. And then finally, we found that asymmetric dispersal led to this two-body problem that increased variation in mutualisms. So, all in all, we find that G by E can maintain the variation that we observe within mutualisms, but when predicting how these interactions might respond to a changing environment, it's really important to understand the ecological context that shapes these interactions, namely rates of dispersal and the relative fitness effects of G by G and G by E interactions. All right, that's all I had prepared for you all today. I just wanted to say a great thank you to my advisors, Matt Osmond and Megan Fredrickson, for all their help with this project, and members of all the lab groups that gave me help and feedback. And if you have questions, please ask me now, but if you're shy, you can check out our preprint. Thank you.
When you were modeling the, like, kind of like the landscape, you assume that the heterogeneity is constant, but it can vary.
So the question is, can the heterogeneity in our model vary? And do you mean like the changes in fitness between environments or like the number of environments in the model?
Yeah, assuming like a grid
Uh-huh.
with like little changes
Mm-hmm.
in the like environment and its fitness between the host and the environments, or a really heterogeneous like landscape, for example.
I see. Yeah, so our model just featured two distinct patches, but you can envision that as dispersal between those patches is increasing, you have more gene flow between them. So we didn't consider like more continuous or clinal variation in the habitats that genotypes experienced. Yeah, thank you. Yeah.
I like that you call the butterflies symbionts. I'm wondering, you know, with true like, you know, bacterial symbionts or something that are maybe like super tied to their hosts and can't really switch between hosts. Is that a parameter that you could vary and maybe see how that affects how mutualisms do in different environments?
Yeah, that is something we could vary. So the fidelity with which the partner is transmitted along with. And we've thought a little bit about this, at least in the discussion of our current manuscript. And we think that having a partner that's sort of maintained with some amount of fidelity might behave very differently across different scenarios. And I'm trying to think through my intuition there. I think it's that as you have a partner that's maintained with its host, it tends to increase the scope for positive frequency dependence because it's much easier to build up the abundance of a partner that you prefer. Yeah. Yeah.
I'm always curious about how your model relates from a one-to-one mutualism to like a one-to-many mutualism. Do you think that this holds throughout those different like asymmetries and the number of associations?
Yeah, that's a great question. So it's like, how does this relate to like partnerships that are much more diffuse? And I think that I've conceptualized this best for a one-to-one interaction, but it is sort of, we use like a mean field dynamic in that, so within our model, it's sort of equivalent to think of each host and symbiont sort of like each having simultaneous interactions. So you can think of that being as like, you know, one plant that's pollinated by multiple butterflies at the same time. And so like to that extent, we do apply to interactions where they're more diffuse partnerships, but it gets more complicated, right, as you add more and more and more genotypes. Yeah. Cool, thank you.